All right, welcome everybody. Good afternoon. You see a new face here now. My name is Angelica Hilbeck. I spoke on occasion and I think we presented ourselves yesterday morning already, but for those who have not seen me yet, I'm a scientist and researcher at the Swiss Federal Institute of Technology, an agroecologist by training. I've been doing a lot of risk assessments of genetically modified organisms and contributed to the debate on genetic engineering in agriculture and the transformation of agriculture for roughly 30 years now. So I'm very glad to be the person to chair this afternoon session, the last session now. I'm sure you all are very exhausted by all the wonderful talks and discussions we had. But please, I hope you have regenerated yourselves and re-energized, because we have two exciting speakers still waiting for us in this afternoon session. And it's interesting also to see that we have started out with genetics or critique of current concepts and understandings of genetics yesterday, then took a long way through other topics and issues and are now coming back again to a critique, as I understand it, of the current understanding of genetics and biological or genetic causality in it, to touch on issues of endocrine disruption, but also a topic we haven't discussed on with our second speaker, the epigenetics of the concept thereof in inheritance and evolution. With that, I would like to get started. I'm trying, I hope I can master the technology around me here well. I'm assisted by everybody else who has much more experience in doing this. And I would start by introducing our first speaker, Ana Soto. I think, Ana, we met at least once, probably twice, I think. And so it's good to see you again, good to meet you. I wish you all would be here in the room. But at least we can do it this way now and see your pixelated self at this point, as a friend of ours always calls us. Let me briefly introduce Ana, and I had it all prepared. Yes, Ana Soto is an MD and a theoretical and experimental biologist. She's a professor at Tufts University School of Medicine and a foreign correspondent member of the Center Cavalier. I'm not going to pronounce this. It's the ENS in Paris. That's where we met, I think, when I gave a presentation there. And she's a fellow at the Institute of Advanced Studies in Nantes. Her research interests include the control of cell proliferation, morphogenesis, and the developmental origins of adult disease, and theoretical and epistemological topics that pertain to biological autonomy. And that is what is driving her research and her motivation, I guess, to accept our invitation to speak about biological causality and the application to endocrine disruption. Ana, I hope I didn't make any mistakes. If you want to add, please do so. The floor is yours, I would say. The virtual screen is yours.
Thank you very much, Angelica. And I would like to thank the organizers for their invitation to this, to talk here, and to the excellent meeting. It's just a pity that I'm in the States, so people that spoke at 9 in the morning, obviously I was sleeping. So I probably would repeat things that you have already said at that time. But I just couldn't join you in Bern, which is one of, I love Bern because I worked for 5 years with the Swiss National Science Foundation. Okay, now let's get into matter. So what I'm going to do is I'll start presenting the present situation in biology. And I think that here you are very critical, so probably I won't add much more to what you already know, but I think that this is necessary in order to do the next thing, that is to propose a way to get out of this impasse, and stressing the necessity of theoretical frameworks in order to do research in biology, And then, and only then very briefly, I would relate this to endocrine disruptors, because I think it's more important at this point to get into what type of theory we need in order to study biology or do research in biology. So I will start with what we consider the impoverishment of biological thought. Karl Marx developed the concept of proletarization as a process where people move from a situation of unemployment or self-employment to one of being hired as a wage labor. And at that time, because of the rapid industrialization, these people were put in the chain of production process, in doing just one part of an object, and therefore being compelled to reduce their know-how. And this is proletarianization. Now, unlike proletarianization of artisans, that is what Marx described, the proletarianization of biologists didn't start by introduction of techniques, but it was, on the contrary, started as a problem in theory. This conceptual impoverishment started with the idea by biologists that their discipline could be reduced to chemistry and physics, and that cells and organisms are analogous to machines. And part of this problem started with von Helmholtz, that, by the way, was a physician, but he became an excellent physicist. So there is where the trouble started regarding the conversion, not of humans to machines, but the reduction of biology to physics. Okay, then in turn, this led to the idea that causality is to be found at the molecular level in genetics and genes in particular, molecular level in general, and genes in particular. So we started about 2 centuries ago with this problem. So this conceptual shift became more acute at the turn of the 19th into the 20th century, when agency and normativity hitherto consider core characteristics of what is alive. So those are properties of organisms. Those properties were transferred to natural selection. In other words, when the theory of evolution became to be accepted, and this is not Darwin's actually, but their followers, that gave so much weight to natural selection that took all these properties of the organism and transferred that to natural selection. The organism became a passive, inert thing, if you will. So this is the beginning of the ascent of genetics and of genetic determinism. The abandonment of the classical concepts that distinguish biological entities, and of which I will talk later, agency, normativity, and goal directness, was accompanied by choosing mechanism as a privileged explanatory and causal regime. Much later, and now we are talking about the 1940s, the advent of mathematical theories of information and the advent, of course, of computers further transformed biology. The organism and its cells are now seen as a program and the product of information that is carried in the DNA and transmitted downstream. So I think that this explains much of the problems that Siguna Müller explained to us yesterday, because of this concentration in an extreme molecular and genetic reductionism. Trouble is happening everywhere. And one of those problems is the stagnation that we see in the understanding of biology. The consequences of this conceptual shift, I would say, are foremost the adoption of information as a reified metaphor. Nowadays, most biologists are convinced that theories are not needed, that they really absorb the world. So they think that you just can see reality, and they ignore what is obvious to philosophers, that science contains a philosophical baggage, and their problem is that they are taking it on board without examination. So therefore, history and contingency are forgotten, and the role of these two things in biology. The becoming of an organism, which is normally, should be thought as being played at least in 4 dimensions, is reduced to a linear program. And this ignores the complexity of multi-level interactions of cells, extracellular matrix, biomechanics, bioelectricity, just to become a play of molecules orchestrated by information emanating from a program contained in DNA. So molecules are elevated to contain a privileged role in causation, particularly the molecule of DNA. And then, of course, what I just said in the title, there is this reification of the idea of information. They think that it exists in DNA, and therefore is what is governing those obedient cells. Okay, how do we get out of this? Well, I think that we should, for a minute, review the aim of science and go from there. So the aim of science is to seek knowledge for the understanding of nature, obviously, and also for guiding us to act responsibly when using this knowledge. At the beginning of the scientific revolution, scientists thought that they had direct access to their outside world, and thus that to the objects of study. So actually here I'm referring to Descartes, because he thought that goodwill was to make nature intelligible to creatures like us. But then, in the 19th century, the separation of science and religion removed this simple idea from our minds. And so we had to, scientists had to acknowledge that they are inside the world that they wish to observe. And that poses a problem, undoubtedly. How do we access the objects that we would like to study? And so I think that I would go through a little detour using an example provided by von Uexküll. And he studied the tick, which is an insect that is blind and deaf, but that it recognizes very efficiently its prey because of the smell of its skin. And once the tick falls on its prey, which happens to be a mammal, the warmth of the skin signals that he's there. So the tick inserts her head into the skin and sucks the prey's blood. So we know that. But so what is the word of the tick? We see that the tick is just guided by gravity, by olfaction, and by temperature. Obviously, its word is very different than ours. So the fact that our word is bigger in the sense that we have 5 senses, does not imply that it's more complete than that of other species. So right there is the problem. So we need tools to access the objects and provide objectivity. And theories are the tools that provide this objectivity. So that is the reason why we need theories. And scientific theories provide organizing principles, and with them construct objectivity by framing observations and experiments. And the theory determines which are the proper observables. And I think that that was discussed very briefly by Thierry Paul when he referred to Einstein. Conversely, the choice of observables is a major theoretical commitment. And these two things, we can say it for people that are not in the thick of theories. Newtonian theory doesn't allow you to see, to take an atom as an object of study. It's out of the constraints of that theory, and so forth. But we are interested in biology, so we have to look at physics as a place rich in theories in order to understand how theories are helpful. That doesn't mean that we have to transport theories from physics to biology. And actually, I would say that a theoretical transition is required to move from physics to biology. So the first thing that we can copy from physics in a way is to find the limit case and define it. That in Newtonian physics, that will be inertia. And in biology, we call it the default state, more to follow later. The awareness that biology is about change. And thus, two very important aspects of physics do not apply to biology, which are a predetermined phase space. That is something that Giuseppe and Mael have worked extensively on it. And the second, that invariance and symmetry conservation are also not as important as in physics, since biological beings are breaking symmetries at every second. Additionally, we have to think that organisms are historical, so that is very important, and they are changing. And biological objects are goal-directed normative agents, that they try to keep themselves alive, that they have a capacity to generate action, and that they are normative, that they create their own rules. And we are just going to show in a minute an example of that. And I will also say that in biology, there is an interdependence between the organism and its parts. So it's a sort of circular causality that we have to take into consideration. And here we have a little example of normative agency. This is an animal that was born with paralyzed anterior members. And this goat, see that it walks, it's not jumping, it's walking, it's a biped. And she arrives at it, and she survives, and she eats, etc. Because she, I mean, not everyone can do it, had the capacity to create her own rules. And those rules allow her to change the bones of the pelvis and the insertion of the muscles that maintain biped station in a way that makes her closer to humans than to goats. So this is just to give you an idea of what has been erased pretty much with all these transformations I talk about that happened during the last 180 years. So that brings us to explanations in biology. We prefer, instead of using cause, we prefer to use Giuseppe's phrase structure of determination to talk about how a phenomenon is determined. The organisms that we studied actually are full of causes since at least the last 3 billion years, because at that point our common ancestor appeared on Earth. And so causes come from long ago. Explanations are not exclusively causal. So we can explain that the heart functions is to pump blood. That is a sort of why. And also can provide a causal explanation to address how it does it. So sometimes people confuse that a teleological explanation with a causal one. In spite of their dominance in biology, mechanisms are not the best way to address biological causality, since the relation between the organism and its parts is that of a circular reciprocal interdependency. Which brings us to a particular philosophical stance that is called organism. That was the first wave of organicists to appear at the beginning, 1920 or so, and they propose the centrality of the organism concept in biological explanation, the importance of organization as a theoretical principle, and we are going to talk of that later, and the vindication of the autonomy of biology. So that is that the biology cannot be reduced to physics and chemistry. Additionally, they made clear that biological explanations are not reducible to mechanical ones, as organisms are not machines. And the distinctiveness of organisms can be understood as a consequence of their self-organizing and self-regulating capabilities. But here we shouldn't forget that organisms are historical. So this self-organization is coming from a previous organization of their parents. It's not like this spontaneous self-organization phenomena that we can see in physics, like Bernard cells, etc. In 2013, I became the Blaise Pascal Chair at the École Normale Supérieure, and my host was Giuseppe. And the goal of this project of mine was to identify principles for a theory of organisms, but obviously it wasn't just a project of myself. It was a group effort. We were a group of 8, of biologists, philosophers, and mathematicians and physicists. So Carlos Sonnenschein, Mael, Giuseppe Longo, Paul Antoine Miquel, and new people like to our group, like Matteo Mosio, Arnaud Pocheville, and Nicole Perret. And so we published an issue of Progress in Biophysics and Molecular Biology in 2016, where we talk about this elaboration of principles. And we continue. This is just to say that now I'm going to talk of those efforts. The first thing is the notion that we arrive at, Carlos Sonnenschein and I, in the early 80s. And the default state is what happens when nothing is done to the intended object of system. So the default state in mechanics is inertia. If no causes like changes in energy or momentum modify the properties of a physical object, the object conserves its properties. Okay, so this comes back to Galileo. And then our default state in biology is that the unconstrained state of cells in the presence of nutrients is proliferation with variation and motility. This is rooted in the cell theory and in the notion that cells are agents that generate their own changing rules. So at the beginning, Carlos and I were talking about proliferation and motility, and in a paper that we published with Giuseppe and Mael, we extended variation as also a fundamental property of the default state. Now, the default state, in a way, relates very well with Darwin's ideas of evolution as descent with modification. And it's a non-conservation principle for phenotypes. Now, Darwin made explicit the concept that living entities reproduce following a geometrical progression. And here is how he put it on paper. There is no exception to the rule that every organic being naturally increases at so high a rate that if not destroyed, the earth would soon be covered by the progeny of a single pair. And here in a little diagram, I want to say that actually the cell theory and the default state are a link between the evolutionary theory and the theory of organisms, because the default state of cells applies obviously to evolution from unicellular to multicellular organisms, and also because it applies to a theory of organisms, because organisms can be just unicellular, or if they are multicellular, they arise from a zygote that happens to be a single cell, a fertilized egg. So to conclude with the default state, I have done a little error here about how to order those things. But anyway, our proposal has straightforward consequences of what requires an explanation in the sense of a theoretical cause. And like inertia, the default state doesn't require such a cause. But to the contrary, a departure from it, so cells that are quiescent instead of proliferating, cells that are not varying, or cells that lack motility require a causal explanation. And the default state, proliferation with variation and motility, expresses agency and modifies the causal structure with respect to the theory of the inert. So the second principle, that is the principle of variation, I got stuck here. Oh, good. Extends to Darwin's notion of random variation. In physics, as Giuseppe and Mael say, objects are generic and evolve in well-defined phase spaces, where in biology, objects are specific and the phase space is not set a priori. And that said it in a very basic way, is that in physics, or at least in Newtonian physics, an apple and a planet are generic objects. You can interchange them in a sense. However, in biology, we are individuals. Objects are specific. The principle of variation is expressed in terms of symmetry changes. And the relentless change addressed by the principle of variation points to the major difference between the theories of the inert and those of the livings. By the way, this principle of variation is obviously a source of novelty. And the consequences of this principle are the historicity, contextuality, variability of biological objects. And just to illustrate variation, you can see it in here. This is the mammary gland of a mouse fetus. Actually, each one of these dots is one mammary gland. 3 days later, from that dot, a whole tree appeared that I'm showing here. And you can see that the shapes of those trees are very different. That is the individuality of each mammary gland within the same mouse, but also the variation that represent differences among individuals. Contextuality, we see here how the rigidity of a matrix, the matrix is what surrounds cells in brief, would be one of the ways to specify phenotype. So in a soft medium, and this is the same cell type, would give rise to a neuron. In intermediate rigidity medium, it will give rise to a striated muscle cell. And in very rigid milieu, it will give rise to a bone. So this idea that you need signals that are molecules is not applying particularly to this type of specification. Contextuality also appears in during embryonal development as a big determinant of phenotype. So here we have two organs that have two components. One that is the epithelium, that is what's considered the functional part of it. The mammary gland, the epithelium will eventually produce milk. That is why it's the functional aspect. And the salivary gland will produce saliva. But if you separate, and this is the stroma, where those epithelia are embedded. So if now you dissect the stroma from the epithelium and recombine it, you will see that here the salivary gland epithelium acquires the shape of a mammary gland epithelium under the influence of the mammary gland stroma, and vice versa. So much for a deterministic Laplacian program. And I think that this has illustrated what I wanted about contextuality. We will go then to the third principle of for a theory of organisms, that is the principle of organization. And the principle of organization provides a source of stability and robustness, and is the fundamental source of this stability. And here the important thing is the notion of closure of constraints as a means to achieve and maintain stability. That was at the beginning applied to unicellular or intracellular processes, but now it has been working very nicely for metazoan organization. So in here, we can distinguish two things, the processes and the constraints. The processes are physical and chemical changes that occur in biological systems. They involve the alteration, consumption, and or production of entities. And constraints are entities which, while acting upon these processes, can be said to remain unaffected by them, so they last longer. And here is a little scheme that you don't have to learn it. It's simply to show here, I mean, the squiggles are the constraints, that through this closure of constraints, you are constraining processes in a way that everything in this in this schema becomes intertwined with the others in an organized way. So this is a very important principle, and in this version even more important, but remember that it was about the basis of the idea of organism. But this is the modern version, and we think that, okay, you can read about that in Museo, Monteville, and Longo. And then I want to talk about another interesting phenomenon in biology that we have to explain, or at least take into consideration, and that is level entanglement. So the zygote here is both a cell and an organism from the beginning. And you see as a development continues, so from one cell we get 2, 4, etc., etc. So the idea that an organism is formed by cells that relinquish their independence is inaccurate. I mean, those cells come from an already existing organism. And so their relationship is very intertwined and has to do with how the autonomy of the cell is controlled when it's within an organism. Just to summarize now how you apply these principles, organogenesis, we can see it in general as a process whereby the default state results in the growth by generation of new cells and the production of extracellular matrix by those cells. Shape of the organs and the organism is a result of this default state and the set of constraints, like, for example, exogenous to the organism, like gravitation, and the constraints created by the living organism, like muscular tension. And the ability of the organisms to generate new constraints at each new iteration generates diversity, given that iterations are not identical. Because from the very beginning, a cell, when it divides, the two cells that result from it are not exactly identical. They are already slightly different. And so that brings us back to teleology, something that we briefly discussed a long time ago in the relative time of this talk. And I want to say that because yesterday, I added this one to the talk, because yesterday, Diederik and others were discussing teleology. And I would say that anti-teleological arguments are inconclusive, and because they entangle things and that are not exactly the same. Like, for example, the assumption that every explanation must be causal is not necessarily true, because you can explain, for example, that the heart's function is to pump blood, and that doesn't imply that pumping blood is the cause of the existence of the organ. So you can explain something by what it does without having to claim that what it does precede what it is. The intentionality argument is also a bad argument, because it's assuming that in order to have a goal, you need to be a rational agent. And in any case, I guess that you have seen very primitive organisms exerting goal directness, and they are not rational, so there is the proof. We need to invoke the capacity of organisms to pursue goals in order to explain the origin of adaptive novelties. And those arguments I borrow from Danish Walsh. But also teleology could be a causal regime. And here I'm borrowing from Mosio and Beach, in the way that biological systems are, in a way, I mean, the slogan is biological systems are what they do. So self-determination implies a circular relationship between causes and effects. The organization produces effects, for example, the rhythmic contractions of the heart, which in turn contribute to maintain the organization. Like the cardiac contractions enable blood circulation and thereby the maintenance of the organization. So closure is the organization of a set of mutually dependent constraints. Closure does not imply an inversion of the temporal relation between causes and effects, since it consists of a purely relational description of the circular causal regime at work. So biological organization is teleological because it realizes self-determination, that is self-constraint. Self-constraint implies that the circular organization specifies its own dynamics. That is why only self-constraint involves self-determination, and why biological organization, by the realization of closure of constraints, is teleological. So now we go back to the third principle of the three that we identified for a theory of organisms. That is a principle where teleology could be perfectly naturalized. And now I would just talk very briefly about our other field of inquiry, that is endocrine disruption, and the relevance of this take we have on causality to endocrine disruption. So regulation, we feel, after 30 years of working on this, that is moving on geological time speed. So we are very frustrated. And the causes are diverse. One is the relationship between science and society, where you know what the actors are. There is conflict of interest, there is lobbying, there is this new field of inquiry. It's amazing that we scientists have to work defining what is agnotology, the science of studying the creation of confusion and ignorance. And we also have to face an outdated regulatory science. So those are things that you already know. And then there are the problems inside the science of endocrine disruptors and endocrinology, which is a theoretical crisis, the one that I described, because it permeates every aspect of organismal biology. And then there is the other issue of science for the sake of science versus applied science. There we are making progress, I should say. But on the theoretical crisis, we are far from fixing it at this point. So one of the things that affect us at every minute when we deal with the industry is that the industry always says, oh, you don't have the mechanisms, go back to the bench to find, and we cannot regulate if you don't show the mechanism. This is totally unnecessary, because you can describe effects without, you know, in a black box, you put something in and something comes out. So, well, that's it for these regulatory purposes. But they insist on that you have to get to the mechanisms, and people that work on regulatory science, of course, go along with that. So this call to do more research is appealing to experimentalists, particularly to those that adhere to reductionism and want to get more molecular detail. And you know that this is a bottomless pit, because mechanisms as such don't exist, and you will never find the single molecule that is producing the mechanism, given that, as I said, causes are acting for the last 3 billion years, in the sense that we all descend from Luca. And the endocrine disruptor community is now aware that there is sufficient evidence for regulatory action, because we see those effects and we can quantify that. Now, regardless of one's theoretical preferences, now we can discriminate between science for its own sake, in which time is not of essence, and applying science to the benefit of society. So we have arrived up to that point. However, there are some issues that have been discovered by these scientists during the last 30 years, like non-monotonic effects of developmental exposure to endocrine disruptors. And by that I mean that instead of having a linear relationship, that the more chemical you put, the bigger the effect is, doesn't apply. Some endpoints are sensitive to very low doses and insensitive to very high ones. And that can be perfectly explained, but I'm not going to work on that right now, because I have to not talk too long. And we know that I can enumerate all the types of effects that a single chemical is producing in animals: cancer, decreases of fertility and fecundity, increases in altered behaviors, obesity, metabolic syndrome, etc. But there are other things that were discovered, and yet we have to find a more detailed explanation, and it's that there are transgenerational effects that are not mutational. So that is that transmission of phenotype can occur in the absence of changes in the DNA, which is okay, it's an epigenetic change. And I think that we are stuck because of this reductionism and genetic reductionism around us to really find appropriate explanations. There are additive effects of multiple exposures. That is much more easy to explain, and it's been done. And the lack of explicit and rigorous theory frames has consequences, because, again, we are told to look for mechanisms, and there is a lack of understanding of the specificity of individuality. And because each individual may react differently, and of the contextuality of these effects. And there is a devalorization of observations that don't fit into the mainstream set of beliefs. Just to illustrate, to finalize, I think that there is light at the end of the tunnel, because the European Commission came up with the chemical strategy that I think is rather good. I mean, I think it makes big changes to what we were used to. And this is a great step forward that is the result of the efforts of scientists, journalists, politicians, and medical scientific societies like the Endocrine Society, of which I am a member. But meanwhile, you know, the usual suspects are already creating confusion, ignorance, and we are at a point where endocrinologists are really ready to oppose these delaying tactics by just saying enough is enough. And meanwhile, this is just 1 week ago, the French Agency for Food and Environmental and Occupational Health and Safety, well, it was found that they had hidden a report on the glyphosate. So big scandal. And next scandal, this is in Europe. The European experts that are working on the reauthorization of glyphosate excluded 99% of the academic studies on this chemical. So you see that there is a big problem there, but I think that it is urgent to develop proper theoretical principles to provide a framework for experimentation and for explaining the phenotypes arising from endocrine disruption, and to block the efforts of the merchants of that. So I think that we have work ahead trying to get to a theory, because when you have a proper theory, it's much more difficult to create confusion and ignorance than it is today. Thank you for your attention, and special thanks to the organism group of that I briefly summarized their findings, and to the organizations that funded our work. Thank you for your attention.
Thank you very much.